In Chapter 3, we're going to continue our discussion on proteins, but this time we're going to look at how we actually study the proteins. At the end of Chapter 2, we did start to get into how we can denature the protein, and that will allow us to look at that primary structure, or ultimately determine that primary structure. So we briefly hit on the different types of reagents we used to break down that overall three-dimensional structure and start to study the primary structure. In this chapter, we're going to look at a little more of the details that are associated with protein structure or understanding proteins. So some of the goals that we have for this chapter, we do want to go through and identify and even understand the methods that are used for protein purification. We want to understand and look at that process of protein denaturation. So we'll give a little bit more detail here than we did in Chapter 2. We will talk about the different types of instrumentation that are used when we're analyzing proteins. And we'll even talk about how proteins can be synthesized. So going the other way, instead of breaking them down, actually forming these protein structures. So when we're going through protein separation, there are some key questions that we want to answer. First, we want to know how the proteins are separated, why we would separate those proteins. In terms of the separation of the proteins, we are going to look at different techniques that will allow us to look at different fractions of proteins. We also want to look at why we would synthesize proteins. What are the reasons for actually putting these structures back together? And then the importance of the proteome. Why is that important? So the proteome, this is just representing the proteins that are expressed by the genome. Okay, so these are all the proteins that can be expressed. And it represents that functional expression of information. Now, the proteome is going to vary depending on the cell type that we have. So different proteins may be expressed in different cells. It also is going to vary depending on the developmental stage. So at different stages of development, you might have different proteins that are expressed. And it will vary depending on the environmental conditions that are present. So getting into this idea of protein purification, there are some general steps that we are going to explore as we move through. First, we're going to talk about identifying the protein itself. And the way that we identify the protein of interest is through an assay. So we'll talk about the assays here in a second. Once we identify the protein or identify the fact that the protein's present, then we want to take that protein and we want to release it from our cell. And the way that we do that is we actually have to disrupt the cell membrane and extract that protein out. So oftentimes what's used is a technique called differential centrifugation. And again, we'll get to more details on that in a second. Once we release the protein from the cell, we have to make sure we are separating out that protein of interest, because there's not just one protein per cell. We have multiple different types of proteins that we have to narrow down until we get to the protein of interest. And then once we think we have the protein of interest, then we can explore quantitative analysis of that protein. So the way that we recognize the protein of interest is through the assay. And there are different types of assays that do exist. Some of the assays that we have will rely on binding of dye molecules to the protein of interest. So when this happens, the dye molecule binds to the protein of interest, and we actually can separate out by the color of that fraction. Some of these assays will rely on binding of metal ions. And some of these assays are immunoassays, which we'll explore more as we move through the semester. But when we are looking at these assays, the things that they're measuring, first and foremost, the most important part is that the assay can tell us the presence of the protein. So it can tell us that a protein is in a sample or not in a sample. But it can also give us information about enzyme activity, because we have proteins, all proteins are enzymes. And so by looking at certain products that are formed, we can get information about the activity of that enzyme. So just an example of an assay that we have here. Here we have an assay. It's showing an assay that functions by measuring enzyme activity. So this is a case where we're not looking at the presence of the protein itself, but we're looking at enzyme activity. In this particular case. So in this case, we have this enzyme, lactate dehydrogenase, and this functions to convert lactate to pyruvate. Okay? Lactate to pyruvate. The enzyme that acts on this is lactate dehydrogenase. We're also producing this NADH, so the NAD plus is converted to NADH, and we have some protons that are here. So the NAD plus picks up electrons, becomes that NADH, so we say it's reduced. In its reduced form, that NADH, this substance here, in its reduced form, it actually absorbs light. And it absorbs light at 340 nanometers. So what we can do is we can monitor the amount of light that's absorbed at 340 nanometers. So something like the instruments that we have in lab, we have the SpectraVis or the Spec20, where we can look at absorbance measurements. You can use a UV-Vis if one is available. But you measure the absorbance of light at 340 nanometers, and that is going to allow you to determine whether or not the protein is present. Because if the protein's present, then you're going to, the protein, in this case the enzyme, then you're going to be producing that NADH. Okay? So if it's present and functioning properly, you're going to observe absorption of light at 340 nanometers. So you'll see a change in that absorption spectrum. So that's the assay portion. And now we need to talk about removing those proteins from the cell. So how do we actually get those out? Well, there tends to be, it's not just a one step and you're done. There are multiple steps that are involved here. So this is just an example of a protein purification scheme. So when you go through this, you disrupt your cell membrane, and then you have a series of cleanup steps, if you will. So you take and disrupt that cell membrane, and you end up with a crude extract, which is basically all the bits that came from the cell. Then you can take that, and in this case, you can precipitate out certain substances. Then you take your sample and you put it through a series of chromatography steps. And remember, the purpose of chromatography is to separate stuff out by components. And each of these types of chromatography will separate based on a different property that's present. Okay, so looking at this idea of removing the protein from the cell initially, the method that's often used is called differential centrifugation. Differential centrifugation. Okay. And that's often used to remove that protein from the cell. So the cell membrane is going to be disrupted. And when you disrupt that cell membrane, you end up with a homogenate, which is just basically all of the stuff from the cell. That then has to be separated, and we separate that by centrifugation. So when you centrifuge something, remember that you're spinning this at pretty high speeds, and the heavier substances are going to come out of solution. So the heavier substances are going to form a dense pellet. Okay. And so we can kind of see that in this figure, this little portion here. These are the heaviest substances that are present. So in differential centrifugation, you're going to centrifuge your sample several times. So the first time, we're going to centrifuge this. The heaviest stuff is coming out, and that first one And here we're looking at kind of the speed at which we're allowing this centrifugation process to occur. Initially, what comes out is the nuclear fraction. Okay, that first pellet is the nuclear fraction. So we know that doesn't have the protein of interest. What we do is we take the supernatant, which is the liquid that's above that pellet, we put that into a new tube and we centrifuge it again, this time at higher speeds. Okay, so after the second, there's our speed there. Again, the larger particles that are present in that sample are going to come out. After that second centrifugation, the particles that are coming out are the mitochondrial fractions. So this pellet that's down here represents the mitochondrial fractions. You take the liquid, the supernatant, that's above that pellet, you put it into a new tube, then you're going to centrifuge again at even higher speeds. That's going to result in an additional pellet coming out. That pellet there is going to represent microsomal fractions. And then you'll end up with the supernatant, which in this case will be cytoplasmic, or the cytoplasm, which will contain the soluble proteins. Okay? So you're going through a step-by-step process. You're yielding several different fractions. With each, you're narrowing it down to remove some of the bigger substances that are present in the cell so that you can focus on a sample that has less stuff in it. So that allows you to more easily be able to identify your protein. So after we get this centrifuge down, we have to go through some subsequent steps for purification of the protein, because that cytoplasm has all of the soluble proteins that are present, and we don't really know where the protein of interest is. So these methods will allow us to narrow down those proteins. So we'll explore each of these individually here in a second. But some of the methods of purification include salting out, dialysis, gel filtration chromatography, ion exchange chromatography, affinity chromatography, HPLC, high pressure liquid chromatography, and gel electrophoresis. Okay, and again, the different types of chromatography are going to separate based on different properties of the substance. So we can first look at this idea of salting out. Salting out utilizes the idea of solubility. So what happens is most proteins are going to be less soluble at high salt concentrations. So what we do is we add salt in different concentrations, and as we're increasing the salt concentration in our sample, we're actually causing the proteins to precipitate out of your solution. Okay, so this method is used to fractionate proteins. Different proteins will salt out at different concentrations. So you have different proteins here than you would here. And if we're looking at this graph, what we're looking at, this axis here, we're looking at protein solubility. Solubility. And on the x-axis here, we're looking at salt concentration. So we can see that as we're increasing the salt concentration, we're actually decreasing that protein solubility in your sample. So this is useful for concentrating dilute proteins in solution. But oftentimes, this has to be followed by dialysis because we've salted out, we've put extra stuff in there, and we have to remove that salt. So the next step would be dialysis, and that's a method that's used for separation, which involves movement of small molecules across a semi-permeable membrane. So we're using the semi-permeable membrane to separate out different substances. So this is a useful method for removing the salt after that salting out process. The larger molecules will end up staying within the membrane, and we see that here. These are our larger molecules, which stay inside that dialysis tubing. The smaller substances you're going to see will end up on the outside of that semipermeable membrane. While this is a useful method for removing that salt, it's not going to allow us to distinguish between the proteins that are present. All of the larger substances will be found in the middle. These may be, you may have several different proteins that are within this. So there are other methods that you need to use in order to further separate out these. And this is where our chromatographic methods are going to be useful. So the first of these we can look at is gel filtration chromatography. And the gel filtration chromatography is going to separate based on size. Okay, so this separates out based on size. This method you may have seen rather than being referred to as gel filtration chromatography, it's also called molecular exclusion chromatography. But they're the same technique. Both of these are separating based on size. So what we do is we use these polymer beads. So gray here, we're looking at polymer beads that are present. We put these polymer beads in a column. The polymer beads are usually about 100 microns in size. And then what happens is the smaller molecules are going to enter the beads and the larger ones can't. So the smaller ones kind of get stuck in there. The larger ones can't enter those beads or interact with those beads. And so the larger molecules are just going to pass through the column. And then as you are allowing your column to run, so you're taking your mobile phase, if you will, and you're passing it through your column, which is your stationary phase. And as you add more and more, you're going to start to see the separation. So you're continuing to add solvent to your column. And you begin to collect fractions all along. So you have these test tubes that you're going to fill. And each test tube might have a different substance in it based on the way they separate. Okay, so you take these and you separate them out, and this is going to be based on size. Okay, so the fractions are collected, and those different fractions can be further analyzed in order to determine whether they have the protein of interest in it. Now that would be where the assay would come in handy, or some other technique to determine that. Another type of chromatography is ion exchange chromatography. The ion exchange chromatography, this is going to separate based on charge. Okay, we're separating based on charge, net charge. So salt solutions of varying concentrations will be used to get the proteins to move through the column. So that mobile phase, that's going to be different salt solutions that we're moving through. And these solutions are placed in the eluting buffer, that mobile phase. Now there are different types of ion exchange columns that we have, and the type of ion exchange column will be determined by the type of protein that we're looking at. So we have cation exchange, and we have anion exchange columns. The cation exchange process will utilize negatively charged beads. We have these negatively charged beads here, and so when your proteins come in, you're going to have the positively charged proteins that are going to interact with those negative beads. Other proteins will move through. Once bound to the beads, you change that salt concentration in your eluting buffer, and it's going to help release or move the positively charged particles through that column. So the less positive the protein charge, the sooner it would elute from your sample. So it will require less of the salt concentration. And this is based on kind of competition of intermolecular forces here as you're adding those salts to your column. And then the anion exchange is going to be the opposite. So your bead would end up being positive, and it's going to interact with the negatively charged proteins that are coming through. Now, we have these substances here, carboxymethyl group, and the diethyl amino ethyl group that's here. And so this is just showing us, these are showing us different substances that would be used for beads. Okay, so for your cation and your anion exchange substances. Next, we have affinity chromatography, and this type of chromatography uses or utilizes the affinity of proteins for certain functional groups. So it separates based on affinity of proteins for certain functional groups. So again, the column has these beads, and these beads that are present in this case are going to be modified to have some substance on the surface. Okay, they have some functional group that's present. Now that functional group will interact with the protein of interest. Okay, so we know a particular protein likes to bind a particular functional group. So we put that functional group on our beads, the protein will come in and bind. So you take a mixture of proteins, you add it to your column, you have a buffer that's run through. The buffer that's run through is going to remove anything that's unbound. So if the protein that doesn't like the bead is going to come through first, or doesn't like that functional group will come through first. The ones that do bind to the functional group will be bound to that particular bead that's in there, the beads that are present. Then, as you did in the past, you change the concentration of that buffer that you're moving through your column or your mobile phase, and that's going to help wash your sample out. So you actually, when you're washing your sample out, that buffer that you're putting in is going to have that same kind of functional group. It's competing, that same chemical group. You're increasing the concentration of it so that you can compete and kick those proteins off of those beads. So the proteins would be released and move through your column. So it's separating based on how much those proteins like the functional group on the bead that's present there. So there are different types of affinity columns that we use. So the resin is really referring to what type of bead is present. So again, that's going to be chosen based on what's the functional group on the protein of interest that you have, or what functional group will that protein of interest bind. And so you don't need to know all of these substances, but this just shows you an example. So if you're trying to get glycoproteins and glycolipids to separate out, you would use this substance, which is referred to as concanavalin A agarose. That would be in your column. Whereas if you're trying to bind or separate out proteins with heavy metal affinity, you would use this aminodiacetate agarose as your substance in your column. And then we also have HPLC, which is another type of chromatography, that high performance liquid chromatography. And this is essentially an enhanced version of column chromatography techniques. It's going to be more rapid. It's going to produce higher resolution results. And so you would take your sample and you would inject it into your instrument. It would run through a column, and this column, depending on what you're trying to separate, it would be packed with different materials. And as it's running through, it's going to tell you, based on the time that it takes to run through, what your sample is. And oftentimes there are databases of different proteins. So we know based on the peaks, similar to GC, and many of you have done GC experiments, we're able to identify what proteins are present or what sample is present. Another type of separation would be gel electrophoresis. So the electrophoresis will allow us to determine how many different proteins result after each step that we have. So in this process, the gel electrophoresis, this is involving the movement of charged molecules in an electric field. So when you do gel electrophoresis, and I believe everybody in here has done gel electrophoresis, we will do this in lab, but you have your gel and you put it in a gel box and you run a current through it, and it's going to separate based on the charge that we have. It's also going to separate somewhat based on the size of the substances. So the smaller molecules will move more readily through the gels, larger ones are essentially immobile, and then because we have this charge difference, the negative particles are going to move towards the positive end, and the positive particles will stay up towards the negative end. So you can put your sample in and separate it out. Now with electrophoresis, there are different types of gel electrophoresis. What you probably have done more often is the method using agarose gel electrophoresis. And that's what's used when we're studying or separating out DNA. When you're looking at proteins and separating out proteins, we use SDS-PAGE. So sodium dodecyl sulfate polyacrylamide gel electrophoresis is what this stands for. Sodium dodecyl sulfate polyacrylamide gel electrophoresis. So on the left here, we see this sodium dodecyl sulfate substance. What this does is you add it to your sample, and it functions to disrupt nearly all of the non-covalent interactions in your native protein. So it's an anionic detergent. You take this and you add it to your sample, and it denatures your protein. Then you take that denatured substance, then you take that treated protein, and you put it in your gel, and your gel is made of this polyacrylamide. Okay, and the polyacrylamide, we have this blue monomer that's present, and then we have a cross-linker that helps make that material, cross-link. So these are co-polymerized together. And then in this, the small proteins will move more rapidly through the gel, and the large proteins will stay at the top. And so the movement of the proteins within this SDS-PAGE is going to be proportional to the log of the protein's mass. This is done in a similar way that the agarose electrophoresis is. You have your gel, you load your sample on it, you pass a current through it, and it separates. Now usually you put some kind of dye in there that will bind so that you can see where your sample is. So this is an example of a gel that results from SDS-PAGE. And the blue color here is not from the protein itself, but it's from the use of a Coomassie blue stain, okay, which allows us to visually see that protein. So the first column that's in here, this is representing proteins of known weights. So you put your standard in there. And that's going to allow us from this standard and how this runs to determine the different proteins that are in each of these other bands that are present. Now in lab, the standard protein kind of ladder that we tend to use is a kaleidoscope protein sample. And so by running that and being able to measure the distance of the bands there, we're then able to compare or plot this information and compare the distance of proteins or bands in these samples to that in order to determine what the size is. So another technique we can use is something called isoelectric focusing. In this technique, the proteins are separated based on relative contents of acidic and basic residues. So the acid-base properties that are here. So we have a gel of sorts that kind of varies in terms of pH. There's a pH gradient across that particular gel. And the protein will move until it reaches a position within the gel where the pH is equal to the isoelectric point of the protein. Okay? And that isoelectric point is going to be the point where the protein has a net zero charge. The gel we'll have, okay, so that gradient, that pH gradient is there, and we establish that gradient by injecting a substance into the gel, which is essentially a multi-charged polymer. And that allows us to separate based on pH. There's also a technique called two-dimensional electrophoresis. This technique is going to combine the isoelectric focusing with your SDS page. So you're doing separation of both of these at the same time. So the proteins are separated horizontally based on isoelectric point, and then vertically based on the mass. This is going to be useful if proteins are isolated from cells that exist under different physiological conditions. So once we get through and we separate the protein and we analyze the protein, then we can get some quantitative information about the protein. And so when we're getting this quantitative information, there are a couple of different parameters that we use. So we can look at total protein, total activity, specific activity, yield, and purification level. And so we can look at each of the steps in that purification process that we've talked about and evaluate all of these criteria for each of those steps. So if we're looking at the total protein that's present, this is the quantity of protein present in a fraction, and it's obtained by determining the protein concentration of a part of each fraction and multiplying it by the fraction's total volume. Okay, so total protein, we're looking at the amount of protein that's in our sample or in our fraction and multiplying that by the total volume. The total activity, when we're looking at total activity here, we are looking at the enzyme activity for the fraction. And so again, what we look at is the activity of the enzyme in the fraction that we're looking at, that particular fraction we're looking at. And we're going to multiply that by the total volume of the fraction that we had. So you take your sample, you measure the activity in a portion of the fraction that has come through your column, okay, and usually you would determine that by an assay, that activity, enzyme activity by an assay. And based on the results of that, you multiply it by the total volume for that fraction. Then we have specific activity. So our specific activity here. And the specific activity we're going to obtain by dividing the total activity by the total protein. So total activity divided by total protein. Then we have the yield. And the yield is a measure of the activity that's retained after each purification step. And that's done as a percentage of whatever the activity in the crude extract was. And then the purification level, that's a measure of the increase in purity of our sample as we're moving through. And that is obtained by dividing the specific activity by the specific activity of the initial extract. So it's a specific activity of that particular step divided by specific activity of the initial extract. Okay, so whatever the specific activity of the step is, divided by that original. So 9 here, we take the specific activity of this step, and divide it by the original, that original homogenized sample. So these are the parameters that we use in order to quantify the information after each step. And once we get through and we're able to isolate the protein of interest, then we can start to determine the primary structure of that protein. So we can sequence that protein. So you take your original protein sample here, and we have a series of steps that we're going to move through so that we can systematically determine what our overall sequence is. So you have your sample, you can hydrolyze your sample into those individual amino acids. So basically, you are taking your protein, you're breaking all of those peptide bonds, and then you can put that in HPLC and identify the individual amino acids that are present. You can take your overall sample and add specific reagents to it, and these specific reagents will tell you the directionality. So they'll be able to identify that N-terminal end and the C-terminal end of your particular protein. You can go through and add substances that will cleave your protein at specific sites. And so you end up with smaller fractions of your protein, which you can separate. And then you can determine the sequence of those smaller portions. So you go through, when you're using this particular step, you add a protein that will, or you'll add a substance that will cleave your protein into specific fragments. Then you take your original protein and you add a different substance that will cleave it in different places. So here we're looking at cleaving it at sites other than what we saw in this particular case. You can determine the sequence of the resulting fragments in each of these cases. Then you can take them and compare the different fragments and look for overlapping peptides, and that will allow you to help build the chain back together. So you're putting the puzzle together so you can see which portions are present. So as part of that scheme, in that step 2, we said we can add different reagents that will allow us to determine what the N-terminal and C-terminal end are. So the substance or the process of Edmond degradation, which is shown here, this will help us identify the N-terminus. Okay, tells us which end has that amino group on it. So in this method, what happens is that you have some sort of label that you add, and that label will bind to the amino terminal. Okay, that amino terminal residue will be labeled, and we're able to, because of that label, we're able to cleave that particular amino terminal residue from the overall protein structure, and it doesn't interfere with the peptide bonds between any of the other amino acids. So we're able to just take here what we're showing. Here's our substance. We put a label on that attaches to that N-terminal end, and then we're able to cleave that off. And so then you would end up, it's released, then you would end up with a new N-terminal end, and you're able to add the label again, which binds to that, and you can pull that piece off. So it allows us to look at that end in that directionality. So the Edmond degradation we looked at, that'll tell us the N-terminal end. The C-terminal end, we actually will go through and use an enzymatic approach. So you actually have different substances called carboxypeptidases, which these are going to cleave the amino acids at that C-terminal end. And you use different carboxypeptidases depending on what residue is present there. But we can, in general, cleave our protein at different portions, and where we cleave it depends on the residue that's present. So we'd use a different chemical substance there. So this would apply to both determining that C-terminal end, but it also would apply to that fragmenting of our overall protein structure. We said we add substances that will allow us to break our original chain down into smaller fragments. And by adding different substances, we end up with different fragments. So this is just showing us a table of different reagents that we can use, and it tells us where those reagents are going to cut our protein. So using something like cyanogen bromide, what that's going to do is it's going to find all of the methionine residues that are present, and it's going to cut it on the carboxyl side. So it makes those cuts on the carboxyl side. Something like carboxypeptidase A, that's going to look for the amino side of the C-terminal amino acid, with the exception of a couple of these, lysine, proline, arginine. Okay, so you choose a different chemical reagent, and it cuts at different residues that are present there. So you cut your protein into smaller samples, then you can run it through a mass spec and determine protein analysis of this. So you can get different peaks that would correlate to the different fragment sizes that you have. And there are different data banks that will help us identify the residues that are present and the sequence of these particular amino acids. We do have some additional methods for protein analysis. Some of these methods include immunological methods like ELISA, Western blots, fluorescence can be useful. X-ray crystallography can give us some information about the crystal structure that's present. And we can even use NMR to analyze our proteins. And we'll look at some of these methods in the context of other chapters or in the context of our laboratory portion of this course. So again, this chapter goes through the different methods for actually looking at our protein and studying our protein. How do we get it out of the cell? How do we break it down? How do we sequence? Now, one thing we haven't addressed yet is what do we gain by identifying these sequences? Well, we actually can compare the sequences of different proteins, and we can identify similarities, and that will give us some insight into the evolution of species. We can get information about repeating structures. We can identify signals that determine location of proteins. We can use the information to generate antibodies for a protein. We can use the sequence to generate DNA proteins for a particular gene that is going to encode a protein. So it's very important that we have information about these sequences, very useful and practical applications. Now, what we haven't put in here, there's not a specific slide for, is in the beginning we said we were going to talk briefly about how we synthesize proteins. So when we're synthesizing proteins, we actually can make a polypeptide chain using something that's referred to as automated solid phase methods. And basically in this process, you have some sort of support, and that support has functionality associated with it, and you end up bringing in individual amino acids, and they're going to form polypeptide, or not polypeptide, but they're going to form peptide bonds. Okay, so you have the carboxyl end of the growing chain links to your support, and then the carboxyl end of the incoming amino acid is going to be joined to what's already there, and it's going to continue to grow that way. And there are certain reagents that we have to bring in order to activate this process. But this is important. These synthetic peptides, they can serve as drugs. So it's useful to serve as drugs. They can serve as antigens that will stimulate formation of specific antibodies. But they can also provide us insight into the relationship between the amino acid sequence and the overall conformation. So you can make a particular amino acid chain or polypeptide chain and then look at the types of interactions that occur between those side groups. So it gives us information really about protein folding.